So, like what I mentioned earlier, today's session is on epidemiology of African swine fever with emphasis on trade-related risks. And if you recall your program, we are going to really go through and gain the most out of this session so that we can prepare the ground for the risk analysis training, which part of epidemiology. And so I think, like I mentioned earlier, we have to be as interactive as possible so that if you don't understand any area, you can ask a question so that we can go to the next to be answered, and so that we can continue getting the most out of the training. Then, like what you're seeing on the screen, sessions 4 to 6, there's a business continuity in the context of African swine fever, zoning and compartmentalization, applying the OI import risk analysis framework to facilitate trade. And on 7th of December, applying the OI import risk analysis framework to facilitate trade. This is actually the bulk of the training for risk analysis. And I want to assure you it will be very, very interesting and nice that you have chosen to attend this training. So like what I mentioned in the first session is that at the end of the training, all the participants who have attended and participated in the training and through the evaluation will be issued with a certificate. And it's nice that you prepare your mind and to participate so that you can earn your certificate at the end of the training. So that is, it is not to scare you or to shove you off. It is adult learning. It is participatory and very enjoyable to follow. We are going to start in another 2 minutes. I can see we have a quorum. We have 30 participants who are joined today's training. And really, it shows that participants are very enthusiastic to hear what the trainers are going to impart to them today. Impartation requires a lot of participation and interaction. So with that, I would want now to invite to start trying your reactions button. For those who have not introduced themselves, I would want to encourage them to press that button and raise their hands so that they can introduce themselves. You also have a chat box where you are supposed to put your expectations and your questions, and we can check that chat box so that we can progress. I can see Professor Solomon Munywa has raised his hand. I wouldn't want to say he should not introduce himself, but today we had reserved for country participants and observers we had reserved for tomorrow. Because he has raised his hand, I can give him a chance to introduce himself. Professor
No, it's
Munywa.
okay, Dr. I can introduce myself tomorrow, no worries.
Okay, thank you. So the country participants, Zambia, if you are there. I can see Namibia, Nigeria. So just raise your hand so that you can introduce yourself. Zambia is a guest. There is a Tanzania, Seychelles, Mauritius, Namibia. Okay, let me give a chance for Zambia to do the introduction. Welcome.
Thank you very much. Yeah. Okay, thank you very much. My name is Dr. Naoma Wuku. I'm a senior epidemiologist to the Department of Veterinary Affairs in Zambia. I'm passing my apologies on behalf of my colleague. Today is not able to join us. He's not feeling well. Thank you.
Thank you, my brother from Zambia. Another hand. Namibia. Oh, I can see a hand from Lesotho. Go ahead, Lesotho, to introduce yourself.
Hello, everybody. My name is Dr. Balasam Paga. I'm a veterinarian at the Ministry of Agriculture, Department of Livestock here in Maseru, Lesotho. Yeah.
Thank you for that brief introduction. I'm looking around. You can put down your hand, Lesotho. I'm looking around for another hand. Namibia, Mauritius, Seychelles, where are you? Tanzania.
Hello.
We can hear you. Go ahead.
Yes, I'm Kungu Selemani. I'm a veterinary epidemiologist in the Ministry of Livestock and the Fisheries in Tanzania. Over to you.
Thank you. Thank you, Makungu Selemani. Seychelles. Namibia. Mozambique. I can see a hand from Namibia. Go ahead.
Good afternoon, colleagues. I'm Emmanuel Hikufe. I'm an epidemiologist in the Ministry of Agriculture, Water and Land Reform, Namibia. I'm responsible for epidemiology and the import and export control. I have 3 more colleagues here. I don't know whether they should introduce themselves or if I have represented the group. Thank you.
We want to hear their voices. Let them introduce themselves. They raise their hand and go ahead to introduce themselves. The other colleagues from Namibia, you just put up your hand and introduce yourself. Just click the microphone and introduce yourself. Okay, go ahead.
Good afternoon, colleagues. This is Simeon Elago from Namibia. I'm a state veterinarian in the same section as Dr. Ekufe, epidemiology and import-export. Thank you.
Okay, your other colleague can go ahead.
Yes, good afternoon. My name is Tovi Piteketa. I'm a veterinarian under the Directorate of Veterinary Services, Epidemiology. Thank you.
Thank you. We have heard you very clearly. Any other? Mauritius, Seychelles? I saw a chat from Seychelles. We want to hear you say something. Gambia. Gambia has raised their hand. Go ahead, Gambia.
Thank you very much. I would just like to introduce myself, Dr. Usman Sise from the Gambia, responsible for veterinary services, animal health in the Gambia. Thank you.
Thank you, Osman. Seychelles, they had a chat. We want to hear your voice. Okay, I think that does it for today, for the introductions. We have used a whole 5 minutes, and time is very valuable. Once you lose it, you can't get it back. But introductions are very important because one, we also test the systems, the way how the participants are communicating to us. And so we are happy that we have gone through that stage. Now, today's session, the team leader is Dr. Livio Heath. And they are going to give presentations with his colleague, who he will introduce himself. And so with that, no further ado, I want to pass the baton to Livio to continue with the program. Thank you. Livio.
Thank you very much, Thomas. I trust you can hear me well enough. Colleagues, welcome to the second session of this training exercise. It's a pleasure for us to spend the afternoon with you, give you some additional information on African swine fever. By way of introduction, as Thomas said, my name is Livio Heath. I'm based in South Africa at Onderstepoort Veterinary Institute, working in the laboratory. I've been working on African swine fever since 2006. and is currently responsible for the diagnostic laboratory at Onderstepoort Veterinary Institute. I'm also the OIE expert for African swine fever in Africa currently. I'm joined today on this session by a second trainer, Dr. Edward Akoth. Edward probably has more experience in African swine fever than I do. He is based at ILRI, the International Livestock Research Institute in Kenya, and he'll be kicking us off today with the first presentation on the epidemiology of African swine fever, specifically focusing on transmission of African swine fever in African context. Edward, over to you.
Yeah, thank you very much. Thanks for the introduction. As Olivia said, my name is Edward Okoth, and I'm based at the International Livestock Research Institute in Nairobi, Kenya. Yeah, I've been working on ASF for many years now, mainly in the field of ASF epidemiology. And currently we are working on diagnostics and also vaccine development at the institute. So today I'll just share with you my screen. Let me know if you're seeing it or not.
Edward, we can see it, but you can just put it into presentation mode.
Okay. Is that okay?
I'm still seeing it as the normal PowerPoint view, not in presentation mode.
So let me see if I can do that. Just a minute.
Edward, it's now in presentation mode, but you can maybe just go to the top of the screen and click it to duplicate, then it will be larger.
I click it.
At the top of the screen,
Yes.
there will be a display option where you can click to duplicate.
Display settings.
Display settings, and then you can just choose duplicate.
Oh.
No.
Well,
it's on the left side.
On the left side, top
on
left
the
side
left.
of the
Oh,
screen.
okay.
Thanks, very much. There we go. Next one.
Oh yeah.
There we
Okay. Okay. And then duplicate.
Duplicate. Yeah.
It's inactive. Sorry.
That's because you reduced your screen first. You have to go back to the right side, go to full screen, and then go back to the left side. And
let me do it again. Okay. Sorry for that. And where's the share screen? I don't know why I'm not.
Edward, you need to put your screen on full screen.
Okay.
We can't see your screen anymore.
Yeah, so let me give it a shot now. Okay. That's the whatever. Okay. And then, sorry for that. Yeah, I should be okay now.
Okay, now full screen. At the bottom right.
Okay.
At the bottom right, full screen.
F5. F5.
Edward, at the bottom right, full screen.
Okay, let me start again with the, sorry for that. This is, let me put the slide on full screen first and then see if that can work for me. Okay. Okay, yeah, that's fine. Right. Sorry. Let me share. Okay, I should be good to go now. Are you okay?
Okay, now you still need to go to the top left side under display settings.
Yes, and then duplicate. Yes, you're
Yes,
seeing it?
you're good now.
Okay.
Go ahead.
Okay, thank you. Sorry for that. Yeah, so our discussion today, of course, will be on epidemiology of African swine fever in Africa. And our discussion between me and Dr. Livio will emphasize mainly on the epidemiology as it relates to trade risks. So the outline of my presentation will be as presented. I'll just give you a brief about African swine fever, how the virus is transmitted. And the emphasis here is that this transmission process will actually inform in identifying specific areas of risk for transmission of the virus, and especially as it relates to trade. Towards the end of my presentation, I'll give you some highlights on distribution and survival of the virus in the environment and also in the tissues. And then at the end of the presentations, I'll give you some few concluding remarks. So this already, I'm sure you already know that African swine fever is a very lethal hemorrhagic disease of domestic pigs and also the wild boar. And it's caused by a very large DNA virus, as for African swine fever virus, which is very stable in the environment. The signs of the disease will be presented later, how the disease presents itself and how you can recognize it in terms of diagnosis. But what I'll do at the moment is more to give you just a recap of the transmission circles that exist, especially in the context of Africa. So within the continent of Africa, African swine fever virus is maintained in the natural host, which is a soft tick in the species Ornithodoros. And these soft ticks can transmit the disease very efficiently, transstadially, transsexually, and transovarially. But then they in turn can also infect warthogs through a tick to warthog infection cycle. And again, there also exists a transmission that occurs between infected ticks and domestic pigs, and this is the circle that we call tick to pig circle. And then once the pigs are infected, they are able to transmit this virus between themselves in what we call a pig to pig transmission circle. We also have a transmission that occurs indirectly through contact or through contaminated shelter, vehicles, fomites, and the equipment. So what I wanted to emphasize here, that the maintenance of this virus and the outbreaks of African swine fever, and especially in the context of Africa, is because of the circle that we call the sylvatic transmission circle that occurs between ticks and then the warthogs. An area where these kind of circles exist would be called permanently infected areas. And the infection keeps on jumping from this circle into the domestic pigs and causing major outbreaks that we witness. So some of the studies that we've conducted in Africa basically confirms that these transmission circles exist. Yeah, we've isolated similar viruses in ticks and warthogs and found the same in pigs in the same locations. Some work that we did in Uganda, not so far long ago, where we were able to track bush pigs, collar them, and make sure that we track them and see their movements across. If you see the picture highlighted here, the darker side is a forested area where bush pigs are nesting, and the lighter side is where domestic farms are located. And we were able to show that this animal travel up to 15 kilometers on a daily basis into the farms. And this probably could bring a possibility of contact between the two species. So again, in this kind of environment, we were able to isolate a similar kind of virus circulating between the two populations of pigs, the bush pigs and the domestic pigs. So some key features in these circles that I've described that I think also in the first presentation was clearly presented is that within the tick-to-tick transmission circle, the soft ticks generally live in animals' nests in case of bush pigs and burrows in case of warthogs. The females lay their eggs in their hosts' nests. Then virus transmission is transstadial, transsexual, and transovarian. And what is critical here is that these tick colonies can maintain the virus infection for up to 15 months in absence of blood meals, and for years with blood meal. So basically this is to say that this kind of tick transmission is able to maintain the virus for a very long time in the context of Africa. Now, these ticks that are infected are able to infect young warthogs. And once those young warthogs are infested and following viremia, they can transmit to other naive ticks during feeding. So generally, warthogs are asymptomatic to infection, but remain infected for life. And within this species of pigs, we don't see any horizontal transmission, and also there's no vertical transmission, meaning a warthog cannot transmit the virus to another warthog, and also they can't transmit it transovarially in that species. Now, the other feature is in the tick-to-pig transmission. This is through the tick bite. So larvae, nymphs, or adults crawl through the nest to find their hosts. These soft ticks feed very fast and at night, but they don't spend much time attached to the host. This kind of tick-to-pig transmission is very prevalent. Some of the studies that we've conducted in Kenya shows up to 17% exposure of pigs to tick bites, indicating that actually this kind of transmission is very possible, even in areas where we don't have warthogs or wildlife. Now, once the tick infects the pig, then the most dominant transmission that really goes between pig to pig is through the domestic circle, which occurs by pig to pig infecting each other through direct contact. But it also can be through indirect contact through contaminated shelter, vehicles, fomites, and equipment. So here I just wanted to highlight that within the herd situation, once the virus gets into a pig herd, we've done some agent-based modeling situation to just look at what the kind of events that then leads to transmission of the virus within the herds. So transmission is basically enhanced by infection rates that can be influenced by, for example, feeding uncooked swill. It can also be due to either contaminated fomites, and this can be either vehicles or beddings and these kind of things. There are some other practices where dead pigs can be sold and maybe this then ends up being fed to pigs, and this contamination also, and also contamination of the environment, can also enhance infection rates within the susceptible pigs so that they become infectious. Quite a number of animals also go through infection, but they never die, they recover. And these recovered animals remain carriers in the environment. And in my later discussion, I'll further discuss what that would mean in terms of transmission of the virus within pig population. So within this agent-based model, you find that you get an epidemic peak by day 28, and that epidemic can die within 60 days. So you'll find either all your herds are infected, quite a number of animals will die, and quite a number will recover. The other approach that transmission occurs is through herd interaction. This could be farms that are at different locations, and infection then moves from one farm to the next. And this kind of transmission can be caused by a number of things. And what influences transmission of virus in this kind of situation is the rate of contact between infected pigs and then uninfected herds in a different farm. And some of the factors that influence this include knowledge of biosecurity, if farmers have incentive to report and isolate animals, things like pig trade or boar service become a big problem with that also. And another loop that affects in this kind of a situation is when dead pig carcasses are sold. We've had in some situation where dead pig carcasses can be sold and consumed, and some of these also fed to pigs. We also get a situation where animals that have been infected, before they really manifest with the signs of the disease, are sold to unsuspecting traders, and this then ends up contaminating other farms and leading to high contact rates. So there are quite a number of factors and practices that then feeds into this loop and causes transmission between herds, from one herd to the next herd. And in this kind of a situation, when you have higher interaction between herds, either through some of the factors that I've mentioned, then this will result in earlier peaks of the infection in the farms that are affected. But if you reduce this interaction rate, even by as much as 20%, then the disease peak is traded for onset. You find that you have lower peaks, and then the onset of disease peaks is delayed for a while, and this definitely means then you can control the disease better. Generally in this kind of situation, where you're looking at between herd transmission, peaks will increase with interaction that is caused by the other factors that I've discussed earlier. Now, the other factor that then also influences transmission of the virus, especially between herds, is what I'll call the social networks. And so pigs' movement form very complex networks, and they're very diverse. Some of these networks are for purchase of pigs for rearing and slaughter. Others are for agistment. Agistment is basically loaning pigs to relatives to keep when food shortages are there. Others are also boar service, where people keep sows, but when they want to breed, they borrow boars from neighbors. The other complex network is the trader and butcher networks, because these people then move into farms to buy pigs and then sell. And this also leads to increasing contact rates between herds in different farms. Animal health service providers also are culprits in this, especially when they visit farms and they don't observe biosecurity. In some of the studies that we've done in East Africa, looking at these social networks very closely, we found out that buying and selling of pigs occur between people who most likely are familiar to each other. And this is what we call near distance relationship. But it's very interesting that during outbreaks, sale of pigs is mostly to unknown persons who come from far off villages. And what this means that when there's a suspected outbreak in one village, you're likely to get another outbreak occurring several kilometers away from the village. And so this kind of transmissions then occur because of people behavior and the social networks that we have. One of the things that we also observed in looking at these social networks, we realized that the key drivers of far distance disease spread in trading nodes are traders, transporters, and butchers. So if you're looking at risk analysis and the transmission of ASF, these are some of the areas that you may want to focus on. Again, I wanted to revisit this, that a number of times when infection gets into, when a herd gets infected, not all animals actually sometimes die. Those that recover then become carriers. We also suspect that in some cases where there's low rate of infection, the animals are then able to control the disease and still remain carriers. So in some studies that we've done in Kenya and East Africa recently, showed that we have quite a number of animals that are asymptomatic, but we are able to detect the virus in the tissues of these animals. And this can really present as a very potential risk for African swine fever virus transmission. So in the later slides, I will discuss this further, and especially to answer the question, such questions as what is the window within which an infected carrier pig can shed the virus and transmit it into naive pigs, and also whether pork and pork products from a carrier pig can affect African swine fever virus transmission. So a lot of studies are needed to know the role of these carrier pigs in virus transmission, especially what then would trigger these animals to be infectious and then transmit the virus. Some of the other studies that we've done in East Africa have also linked infections with seasonal food shortages. So smallholder farmers tend to sell their pigs when food is in short supply and buy new piglets, either immediately or when food is again available. And so this kind of changes in management and also buying and selling leads into introduction of the infected pigs into farms. And a number of times this is associated with the outbreaks, depending on the season that this is happening. So I wanted also to highlight that, yes, transmission can occur within a herd, transmission can occur between herds and through quite a number of complex networks. Yeah, but we've also witnessed a global spread of African swine fever. So I just wanted to highlight this, that the first incursion of African swine fever into Europe was in Portugal in 1957 near Lisbon Airport. And this was associated with feeding of swill. And this particular outbreak led to the spread of the disease in many parts of the world, including the Americas. But this particular outbreak was mainly caused by movement of contaminated meat products. So the spread from Africa to Europe was through swill, but subsequent spread was mainly caused by movement of contaminated pig products. And the map on the left is basically just showing you the two genotypes that have managed to successfully get out of Africa. The first one is genotype 1 that took place in 1957. And then the most recent one is the genotype 2 that took place in 2007. So as you may remember, the 2007 outbreak occurred in June 2007 and was confirmed in pigs in the former Soviet Republic of Georgia, in the Caucasus region. Source of infection was catering waste, including infected pig meat from ships in the Black Sea port of Portai. So these outbreaks of ASF mainly caused by movement of contaminated meat products and wild movement. So this is what led to the spread of the virus across many parts of Europe, including Asia, and even more recently to major parts of Southeast Asia. But not only does this kind of transmission occur from Africa, we think also that transmission within the continent is also possible. And a good example is the recent case of confirmation of a genotype 2 virus in Nigeria. And such kind of occurrences can be either through shipping or sale of pork products from other countries that are currently infected, especially in Europe and Asia, but also within the continent of Africa that can also occur. But I wanted to highlight here that we've been able to generate some tools so far using whole genome sequencing that can really help us know where any cluster of some of these viruses are. And some of the facts that we know so far, for example, is that genotype 2 isolate from East Africa clusters very tightly with genotype 2 covering the Asian and European continents. And this is opposed to the genetic diversity that we see with historical genotype 2s in Southern Africa region. So this base of information is very critical for us to help us trace transmission of these viruses across the globe. So this tracking of virus is done using phylogeny, derived full genome alignment. And the diagram that you see there is use of similar phylogeny to identify genotype 9 and 10 in East Africa. And this has also really helped us to understand the movement of these viruses across the region. Now, for the case of East Africa, we see molecular phylogeny of full genome of the genotype 9 and 10. They show clustering of these East African isolates. And this basically is a very critical information, because if we find these genotypes, for example, in West Africa, it will definitely help us understand that some transmission took place and could be due to breach of either by security protocol, or it could be due to trade-related issues. Moving away from the transmission side, I also wanted to discuss one topic that is critical, especially when you look at the risk of transmission of African swine fever virus. And this is the distribution and survival of the virus in various tissues and commodities. So, for domestic pigs, after infection, the pigs may shed infective amounts of the virus for 24 to 48 hours before clinical signs appear. And during the acute stage of disease, enormous amounts of the virus are shed in all secretions and excretions, and high levels of virus are present in tissues and blood. Like I mentioned earlier, pigs that survive the acute disease remain infected for several months, but do not readily shed virus for more than 30 days. However, infective levels of virus are found only in lymph nodes, and other tissues are unlikely to contain infective levels of virus for more than 2 months post-infection. So this sequestration of the virus in, for example, lymph nodes could be the biggest risk in either recovered or non-asymptomatic pigs that are already been infected. So in vectors and the reservoirs, for example, in the tick, so the tick colonies can maintain the virus infection for up to 15 months in absence of blood meals, and for years with blood meal. So continuously when the tick feeds, then they can lay egg, and then, and as I had mentioned earlier, the virus is transmitted between the stages of the tick development. It can be transmitted via eggs and all that. So this then is able to maintain the virus for a long time. Wild pigs in Africa are asymptomatic and remain infected for life. And so in this regard also, the fact that they are infected probably pork products from these wild pigs fed to pigs could be a risk of spread of the disease, especially in places where these animals are hunted for meat. African swine fever carrier state has been shown in domestic pigs, where tissues from animals show minimum virus prevalence of about 16%, based on positivity in 4 different PCR assays supported by virus isolation. And this point basically confirming the fact that, yes, carrier pigs do exist, and we need to think about this in terms of how we treat the pork and pork products to avoid transmission of the virus. We've also observed in some of our studies that locally adopted pigs, bush pigs and warthogs, show some stronger signals of selection. So while such extremes are absent in commercial pig breeds, especially from Europe, we see a lot of evidence of introgression of bush pigs genes into locally adopted domestic pigs. And this probably could explain some of the observation we see of just a relative tolerance of the virus in some of our local pig breeds that can then also explain some carrier states that then present as a risk for transmission of African swine fever. So the other thing I wanted to talk about is then how do we inactivate African swine fever virus in the tissues and also in the environment. So some of the facts that we need to understand is that this virus is very resistant to inactivation. The virus is stable at pH 4 to 10 and thus not affected by meat maturation. It requires heating at 60 degrees for 20 minutes to be inactivated. Smoked sausages and air-dried ham require smoking at 32 to 49 degrees for 12 hours and 25 to 30 days of drying to be free from the virus. So pork cured products can remain infectious for several months. The virus can persist in the environment in formats such as clothing, shoes, equipment, and vehicles for several days. And African swine fever virus is in a suitable protein environment is stable over a wide temperature and pH range. It has been shown that the virus can survive in serum at room temperature for 18 months, in refrigerated blood for 6 years, and in blood at 37 degrees for a month. Putrefaction does not necessarily inactivate the virus, which may remain viable in feces for at least 11 days, decomposed serum for 15 weeks, and in bone marrow for months. So this basically just highlights how complex inactivating this virus is. So some of the best known inactivation agents include sodium hypochlorite, glutaraldehyde, caustic soda, and potassium peroxymonosulfate. So just towards the conclusion of my presentation, I just wanted to highlight that it's very important that we understand this transmission dynamics of this very complex virus, and mainly because the disease has a very huge impact. Some of the work that has been done by Professor Mary Lou and her colleagues has highlighted some of these impacts that we've seen over the years of outbreaks that have occurred. And these estimates are an underestimate because most of the ASF outbreaks are underreported, mainly because of the fact that there's currently no vaccination or control or a treatment that's available. So there's no much incentive for farmers to report the outbreaks. So this is very critical that we know these transmission circles so that then we can interrogate and also formulate biosecurity measures based on scientific facts. So to conclude, I just wanted to highlight that the virus is very stable and can persist in host through very varied transmission cycles, as has been discussed here and in the previous presentations. Simple biosecurity measures can reduce the transmission, and especially if these biosecurity measures are based on evidence of some of the issues that we discussed here today. The other thing that is equally important is that farmers need to invest in pig keeping in order to implement biosecurity. especially having good housing and quality feed for pigs. And this then will also generate co-benefits for increased productivity of the animals, get them higher returns, and also reduce other correlated issues like zoonotic infections. I just wanted also to throw this to the audience today that farmers will always act in their own self-interest to implement changes on farm if they trust that benefits are likely to accrue from that. And this definitely requires new knowledge that can influence it. Then they also have the means to make changes. And this means they need capital, maybe new feed resources and information, and more open markets to do this. Yeah, but in conclusion, I want to say that whether or not farmers have taken action to reduce risk of African swine fever entering their farms, if they do experience an outbreak, they will continue to act in their own self-interest by selling their pigs before they die, thus perpetuating ASF transmission, unless they have clear incentives not to. So we need to be thinking of other ways to incentivize farmers, using such things as insurance and also compensation policies. So I'll conclude my remarks with that and invite back Dr. Livio to proceed.
Edward, thank you very much for that comprehensive review. You raised a couple of very interesting points there, which I'm sure the audience will mull over and come back to us with some questions. I just want to reiterate to the participants, please use the comment box to post your comments or your questions. We will monitor that, and after the presentations, we'll provide an opportunity to discuss and answer some of the questions and throw it back to the audience where appropriate. There will also, obviously, if your connection allows, there will be an opportunity for you to ask questions later on after the break as well. So after that intriguing talk from Edward, we're going to take a step back into the laboratory now. I'll present a brief overview on diagnostics of African swine fever, how that relates now that is used for risk mitigation on the African continent. You'll just allow me a minute or two to share my screen quickly. Let's see if I have the same problem as Edward. Sorry, let me just do this quickly. Apologies. So I just want to switch off my video to save some bandwidth. So my apologies for that. Let's go again. Edward, if you can just maybe confirm that you can see my screen in large scale.
Yes,
it's
I
fine.
can. It's fine.
Thank you very much. Yes, as I said, the topic of the talk is ASF diagnostics, but I don't want to go into extreme detail on diagnostics. The purpose of this presentation and more importantly, this training exercise is to look at risks, risk mitigation and risk identification. But diagnostics does play a part in that. And I just want to take this opportunity to run through some of the tools that we use in the laboratory now that relates to disease control on the ground. So Edward showed this previously, just a similar diagram of the African swine fever virus, and you should be very familiar with this. But effectively, African swine fever is a large DNA virus, very resistant to disinfection, as Edward just pointed out to us. As viruses go, it's a fairly complex virus. It codes more than 50 proteins, of 150 proteins, sorry, 50 of which actually form part of the virus capsid and the virus, the infectious virus. Proteins on the surface of the virus are often the targets for antibody-mediated response, and we can use this to our advantage in developing diagnostics, and indeed is used as one of the frontline diagnostics for African swine fever. The protein of most interest is the VP2 or B72 major capsid protein, which makes up the largest portion of the capsid of the virus and is present in all strains of African swine fever and is essential for the replication and transmission of the virus. So that plays a central role in diagnostics. We've also heard this before, both last week from Dr. Penrith and today from Edward. It's an acute disease. African swine fever generally presents as an acute or peracute disease with exceptionally high mortalities between 90 and 100%. But it is also important to realize that not every outbreak is associated with 100% mortality. And especially recently in Africa, we've seen outbreaks of moderate mortalities. These can often be described as subacute infections. And important for vaccine development, which we'll come to in the next talk, you also do have strains that have very low virulence and cause chronic forms of the disease. In terms of the disease development and the dynamics of both antibody responses and viremia, it's also very important to understand this in context of diagnostics. African swine fever presents viremia very, very early in infection. Within 2 to 4 days, you can easily detect virus in blood samples, as you can see on the graph. And that remains constant for at least 30 days, if not longer, with sporadic phases where the virus can still be detected beyond the 30 days. Contrary to that, the antibody response is very different. Antibody responses generally take about 7 to 14 days to develop. That's very standard for virus infection. And African swine fever is not any different in that respect. But what is very important for African swine fever, specifically relating to peracute and acute virulence, is that animals die before they develop an immune response, at least the antibody immune response. So if you're testing for antibodies, the first 2 weeks of the infection, the chances of detecting antibodies in an infected animal is relatively low. And that needs to be kept in mind with interpretation of diagnostic test results. In terms of the diagnosis and the tools that we have available, you can broadly class that into 3 categories. Clinical diagnosis, which consists of clinical signs and post-mortem lesions. Sampling and shipment there is quite important for the laboratory diagnosis. I'm not going to spend too much time on that, but I will go through some of the clinical signs and lesions that we see typically in African swine fever. That is usually followed by molecular testing once these samples get to the laboratory, with the PCR method being the primary tool for diagnosis of African swine fever. This is followed by sequencing with phylogenetic analysis based on the sequences. Edward showed some phylogenetic analysis based on full genome sequencing, but we can also use individual genes to actually trace the spread of the virus. And I'll show you a few examples of that a little bit later. Antibody detection, although the antibody response is only late in infection, still plays a role in diagnosis. We generally use a competitive ELISA for antibody detection of African swine fever, but this is supported by immunoblot assay and pen-side antibody test that I will touch on a little bit later as well. What is very important in terms of diagnostics is the sample matrix that you use. The selection of appropriate samples is critical for the accurate diagnosis of African swine fever. I've listed here a couple of samples that we generally receive, largely in order of the usefulness and accurateness in terms of diagnostics. So in acute cases where animals have died and post-mortems have been done on these animals, or animals are terminated and post-mortems done, spleen, lymph nodes, and kidneys are generally the best matrices to send in for diagnosis. These organs contain large numbers of viruses, and the extraction of DNA from these these tissues are generally quite easy, and most laboratories will be quite proficient at that. Serum samples can be submitted for antibody detection, but also for molecular testing. Although the virus is associated with red blood cells, the virus does, or the serum does contain virus as well, although it is slightly less than what you would expect either in whole blood, EDTA blood, or in tissue samples. One of the main reasons that we recommend serum to be submitted to the laboratory is firstly that it gives you a fairly clean signal when you use it for molecular testing, meaning that you don't have false positives resulting from serum samples as you would sometimes have with EDTA blood. But also importantly that you can use those samples for antibody detection as well. So it minimizes the number of samples that need to be collected from the animals. Obviously, with animals that have died, it's not always easy to draw blood for them. In that case, tissue samples are much better. In case of live animals, we always recommend that serum be submitted for testing. The sensitivity on serum is generally a log factor lower compared to tissue samples. You can also submit plasma samples and EDTA blood, although EDTA blood, as I mentioned, can cause false positive results. Meat juices, specifically in terms of import risk, can be used as well. It's a good source for diagnostics, specific and molecular diagnostics, but the sensitivity of the test would also be lower. And then whole blood can also be submitted if nothing else is available. In terms of clinical diagnosis, I'm sure most of you will be familiar with this, but I'll just run through it very quickly. In terms of clinical signs of disease, pigs generally huddle together to stay warm. You often see that together with shivering. Reddening of the skin, especially on the extremities, on the back, under the neck, and posterior parts. You clearly see reddening of the skin, as well as cyanosis on the ears. In chronic forms of the disease, you sometimes see quite severe necrosis of the skin, especially on the belly, the underside of the abdomen, on the neck, and on the ears. This is typically associated with chronic forms of the disease. On post-mortem, what we generally look for is firstly froth in the trachea from severe lung edema. Hemorrhagic gastrohepatic lymph nodes are typically seen on post-mortem. Hemorrhagic kidneys, as well as petechia or petechiation on the cortex of the kidney is very typical of African swine fever. Enlarged spleens is also typically one of the signs of African swine fever. However, you have to be very careful in terms of diagnosing African swine fever just based on these clinical signs, especially in areas where classical swine fever is present. It is extremely difficult to differentiate African swine fever from classical swine fever purely based on clinical diagnosis, and it is recommended that samples be submitted to the laboratory for confirmation. As I mentioned, our frontline test in the laboratory for diagnosis of African swine fever is molecular testing using the PCR. There's several formats of this test available, both in-house as well as commercially. The principle of PCR, as I'm sure you know, is an amplification, exponential amplification of target genes using a polymerase enzyme and target-specific primers. So on the right-hand side of the screen, you can see a graphic representation of that. As I mentioned, this technique is usually performed on tissue or whole blood or serum samples. And most of the laboratories in Africa still use the conventional PCR for the detection of African swine fever. I show at the bottom of the screen is a picture of the output of a PCR reaction run on an agarose gel. And you will notice there's actually two different reactions running here. We have a diagnostic PCR, which we use to identify African swine fever virus in the sample. And this PCR is based on a highly conserved portion of the P72 gene, conserved across multiple strains of African swine fever. This enables us to detect African swine fever with high reliability, irrespective of which genotype or which strain we are dealing with. The second PCR, shown on the left, focuses on a different portion of the major capsid protein gene, and is slightly more diverse in its nucleotide composition, and allows us to perform phylogenetic analysis on these samples. In laboratories with slightly more resources, the conventional PCR has largely been replaced by real-time PCR. The principle is very much the same as the conventional PCR. The chemistry is just a little bit different. In this case, a fluorescent dye is used as the indicator, which means that you cut down on the time required to run the test. In general, from receiving the sample to having an output on a real-time PCR test is approximately 1.5 to 2 hours at most, compared to 4 to 6 hours on a conventional PCR. What is also important is that several commercial tests are available for the real-time PCR, which has been validated and is accepted by the OIE and included in the OIE manual. On the right-hand side of the screen, you can just see an output of what it typically looks like. So a negative sample will have what is indicated in the blue line here, which is below the threshold cutoff, whereas a positive sample has a typical exponential curve with a CT value, which refers to the position or the cycle at which the sample exceeds the threshold or the cutoff value in the PCR. The average sensitivity of the real-time PCR is 99.14%. This is across multiple validation exercises done by various laboratories, several of them OIE reference laboratories, and the average sensitivity is 98.67%. So it's a highly reliable, easy test to do, but can be relatively expensive. This brings me to phylogenetic analysis. As Edward mentioned, phylogenetic analysis is often used to trace outbreaks, to identify where outbreaks have originated and to trace the spread of these outbreaks. Traditionally, we've used the P72 gene for this purpose, and based on the P72 gene sequence, we've identified 24 genotypes to date. Most of these genotypes occur in Africa, with only 2 genotypes, as Edward mentioned, present outside of the African continent, that being genotype 1 and genotype 2. Phylogenetic analysis should always be interpreted together with epidemiological information. It is important that the phylogenetic analysis only gives you a part of the picture and cannot be used on its own to make inferences on the spread and origin of these viruses. I want to give you an example of how this can be used and the usefulness of it in an outbreak situation. What I'm showing on the slide here is a situation that we've had in South Africa. South Africa traditionally, African swine fever has been restricted to the northern parts of the country. However, in 2016, we had an outbreak of African swine fever outside of our control zone in the central parts of the central and far western side of South Africa. This has escalated to the point that we've had multiple outbreaks since 2016. And one of the questions that was asked is, how do all of these outbreaks relate to each other? And what we have done is we have used the P72 genes and the phylogenetics of that, or the phylogeny of that, to try and identify and link different outbreaks that occurred in our free zone. And what you can clearly see is that you have very distinct clustering of viruses. So in this case, you can see genotype 1 cluster, at least one indicated in blue, a second genotype 1 cluster indicated in green, and a third genotype 1 cluster indicated in purple. This suggests that all of these outbreaks could be grouped into 3 different groups with different viruses being responsible for each of these events. In addition to that, we also had an incursion of genotype 2 into our free zone, indicated in the orange at the top here. So all of this data suggests that there have been at least 5 independent introductions of the virus into the free zone of South Africa, indicating the usefulness of this technique to add information to the epidemiology of the virus under outbreak situations. Unfortunately, in this case, the exact source of the strains remain unknown. The next phase of diagnostic focuses on serological testing. Serological testing is used for large-scale surveillance due to its simplicity and the comparative low cost. Per sample, it's a lot cheaper doing serological testing on animals than it is performing a PCR test. However, you have to keep in mind, as I mentioned before, that antibodies only appear 10 to 14 days after the infection, but do persist for quite a long time afterwards, even several years. Since there's no vaccine against ASF at present, the presence of antibodies are always indicative of prior infection. Now, obviously, that needs to be taken with a little pinch of salt at this stage, considering the situation in Asia, which Prof. Penrith referred to last week with the use of unlicensed vaccines. But generally in the African context, if you do have antibody positive animals, then you can safely assume that those animals were either infected or at the very least exposed to the virus. And as I mentioned before, per acute and acute infections, the pigs often die before antibodies are developed. This just shows some of the formats of ELISAs that you can use in the laboratory. They all follow the same basic principle where you have a target antigen, you have an antibody that you want to detect, and you have an enzymatic reaction with a substrate that reacts and gives you a color indicator that is used to judge whether the sample is positive or negative. So in terms of our direct ELISA, you have the antigen and antibody reaction with the enzyme being directly conjugated. Indirect ELISA, you use a secondary antibody that is raised against the target species that you're interested in here, indicated in green. The sandwich ELISA uses a capture antibody together with a secondary antibody and adds sensitivity as well as specificity to the reaction. And then lastly, a competitive ELISA uses a second antibody to compete against your target antibody to identify a positive reactor. The advantage of a competitive ELISA that it's not species dependent and can be used with several different species, which in the case of African swine fever is actually important. So again, in terms of the type of test that we use and what is available, there are several commercially available ELISA kits across both Europe and the African continent. Most of these have been validated to OIE standard and is accepted by the OIE for trade purposes. The kit most commonly used is produced in Spain, is known as the Ingenism PPA Compact kit, and is a competitive ELISA that can be used for multiple species, including bush pigs, warthogs, and wild boar in the case of Europe. The sensitivity and specificity of ELISA tests is slightly lower than that's the case for molecular testing, with a sensitivity of 88% versus a specificity of 97%. But this is still sufficient to be quite useful for serious surveillance projects. It is important, though, to note that positive reactors on ELISA should be, under ideal circumstances, should be confirmed using an immunoblot test, as indicated at the bottom left of the screen. These tests are generally only performed by reference laboratories, but is supplied by the EU reference laboratory for African swine fever to any laboratory that has a minimum biosafety standard. Unfortunately, these tests use infectious material and must be performed in BSL-3 facilities. So in cases where a laboratory cannot perform the immunoblotting on site, we strongly recommend that samples be submitted to one of the reference laboratories for confirmation of results. It is also important to note that there are pen-side tests available for African swine fever, both molecular as well as serological tests. Of particular interest is the use of lateral flow devices in resource-poor situations. There's still quite a considerable debate on whether these pen-side tests should be used as a frontline diagnostic approach. But there's no doubt that under resource-poor conditions, pen-side test lateral flow devices do have a role to play in combating African swine fever. As an example, these tests are currently being used in the Dominican Republic as well as Haiti to augment laboratory-based diagnosis with a recent introduction of African swine fever into those areas. The last of the diagnostic methods that I do want to quickly touch on is virus isolation. Again, this is only performed generally by reference laboratories. Since you are working with infectious material and culturing the virus, it definitely requires BSL-3 facilities to do it safely. And the virus isolation relies on the ability of the virus to actually bind red blood cells, causing heme absorption. Now, heme absorption can be seen as rosettes around infected macrophages, where red blood cells actually adhere to the macrophage, causing typical red rosettes to form. This is very distinctive of African swine fever, and none of the differential diagnoses, such as classical swine fever, form heme absorption and can be taken as a definitive confirmation of African swine fever. We should also just mention that there are strains, naturally as well as laboratory strains of African swine fever that does not cause heme absorption. So whenever heme absorption is done as a diagnostic test, it should be confirmed either by PCR or by sequencing if applicable and available. The last slide on this presentation that I want to share with you is just a summary of what is available and some of the characteristics of the test. So to start off with the virus detection, we've discussed PCR testing. It takes between 5 and 6 hours. It is extremely sensitive and specific. You can use it across a wide range of samples, including tissue, bloods, and cell culture, but it is quite expensive, unfortunately. But it is the most common method used to diagnose or confirm the diagnosis of African swine fever in laboratories. Heme absorption and virus isolation, it's a tedious technique. It takes between 7 and 21 days to perform. It is very sensitive, or can be very sensitive. It is very, very specific. It uses macrophage, porcine macrophage cell lines, so you do require quite extensive laboratory equipment to prepare these macrophages, but it is considered as the gold standard for diagnosing African swine fever in the laboratory. Something that I did not include in the previous slides is direct fluorescence antibody tests, or FAT tests. Those of you that are familiar with rabies testing will be familiar with this test. It is still being used in some laboratories, but is slowly being phased out in most of these laboratories and being replaced by PCR-based techniques. It's a very expensive test, and some of the reagents are not commercially available. So it is not an ideal test to use for large-scale screening of samples. There is a ELISA available for antigen detection, but the sensitivity is quite low and generally not recommended for use as a laboratory diagnostics. For antibodies, as I mentioned, ELISA being the front line, relatively good sensitivity and specificity can only be used on serum and in some cases plasma samples. It's relatively cheap and can be used as a screening test. If commercial kits are not available in the country of interest, there are in-house versions of this test that can be used, which is also accepted by the OIE. Immunoblotting for confirmation, it's a relatively quick test, but quite expensive and requires specialized equipment. And then, as I mentioned, IFA, similar to the FAT, using fluorescence for antibody detection as well, a test that is no longer used in most laboratories as a frontline diagnostics. So with that, I'd like to thank you for your attention and conclude on the diagnostics. Thank you.
Thank you, Livio, for those two very able presentations you have done between you and Edward. I decided to come in so that you can get a chance to look at the chat box. And I have seen some questions coming in. And if there are questions which you also want to ask directly, we have mentioned earlier that we encourage you to raise your hand. And the two presenters will definitely give the answers. We want to do that because it will be now a discussion. We expect to stop at 20 minutes past the hour. We are about half an hour to there. So I think it gives you a chance to look at those areas and answer the questions. Over to you, Livio.
Thank you very much, Thomas. Appreciate the assistance there. Just a correction very quickly, we're not going to go over to the discussion quite yet. I have one more presentation on the illegal use of vaccines that I just quickly want to run through. And after that, then we'll take a break and then deal with the questions. I've noted the question in the chat room. We'll get back to that. We are noting it down. We'll discuss that during the discussion section. So Thomas, if you allow me, I assume I can proceed with the second presentation.
Okay.
Thank you. Again, just give me a second if you want to quickly share my screen. Oops, sorry. Sorry, my apologies. I just want to quickly stop sharing again and just switch off my camera. I'll share again. There we go. So the second presentation is on the legal use of vaccines. Dr. Pendleth introduced this topic extremely well during the first session, and there was quite a number of discussions around this. So very similar to the diagnostics, I don't want to do a full review of vaccine development in this session. I just want to give a brief overview and highlight some of the challenges that we may be facing from a risk perspective with regards to the illegal use or the use of unlicensed vaccines. And potentially, once vaccines are released for use for African swine fever, some of the considerations that we need to keep in mind when we deploy these vaccines. So again, I'm not going to spend any time really on this slide, just the structure and the way that the virus looks. I just want to highlight the proteins of interest for vaccine development. These are not the only ones, but this has been the major focus of, or the major focus areas or proteins of interest for vaccine development. Again, you have your major capsid protein, which makes up more than 70% of the viral capsid. Two minor capsid proteins, the P54 and P30 proteins, that's been used for subunit vaccine development. And then the CD2V gene that's been targeted for specifically for live attenuated viruses. Another set of genes that are important with regards to vaccine development is the multi-gene families that consist of multiple genes or clusters of genes, and are often responsible or involved in virulence and immune modulation, which has also been the target of vaccine development. This just gives you an overview of what the genome of African swine fever looks like. The area mostly indicated in green is referred to as the central conserved area and is fairly conserved across all African swine fever strains. But you have terminal repeat areas that is where the multi-gene families are located, either on the right-hand side of the genome or on the left-hand side of the genome as clusters. And these genes actually differ quite a lot between strains and can be used to identify virulence or differences in the virulence of different strains, but also, as I mentioned, being the target for vaccine development quite extensively over the last 20 or so years. So why don't we have an African swine fever vaccine yet? And I think, again, Prof. Pendrith articulated this very clearly last week, so I'll quickly go through them again. The first thing is that inactivated ASF virions do not induce protection. In the 1960s and 70s, this was clearly shown. There are still some work ongoing on this, but generally it's accepted that inactivated vaccines produced and the way that most traditional veterinary vaccines are produced will not work for African swine fever. The vaccines developed through serial passage on cell culture that was done in Portugal and Spain in the 1960s appeared to be successful at first, but turned out to cause quite extensive problems post-vaccination, specifically with reactions, and I'll show you some pictures of that later on. And for a long time after this, there was a significant reluctance for the use of African swine fever vaccines, which I think we're slowly getting over now with the new technologies available, but it remains a major challenge and a major concern in terms of vaccine development. Ensuring a safe vaccine is really the first thing that everybody is focusing on. There's no point in releasing anything into the pig population that will cause more problems than the disease itself. We mentioned the complexity of the virus, 160 to 175 genes encoded, more than 50 proteins part of the viral capsid. So it's not easy to identify appropriate vaccine targets. Identifying and making sense out of all of these genes and their functions are very, very difficult. And to be honest, most of the genes, we do not know exactly what their functions are. Also, neutralizing antibodies are not effective. Again, comes with a slight caveat. We do know that neutralizing antibodies do play a role, but on their own, they are not effective in protecting pigs from challenge. So some of the newer vaccines aim to combine both antibody response as well as a cellular response to protect pigs against challenge. We've mentioned the genetic diversity. There are at least 24 genotypes described. There's a lack of cross protection generally between vaccines and field strains. So producing a vaccine against the genotype 1 does not mean that you will get protection either against other genotype 1s or against any of the other genotypes that are circulating in the area. This is particularly important for Africa, since all 24 genotypes are known to occur in Africa, and figuring out what the best way is to ensure that all of these genotypes are covered and the different strains and variants are covered by a vaccine is essential before vaccine is applied in an African context. Linked to that, as we don't know what the correlates of protection is, there are some indications, and we do have some idea on this, but really in terms of detailed understanding of the correlates of protection, we're still a long way off there as well. So what are the vaccines that are currently being worked on? I've just highlighted some of the major ones that are being considered. Live attenuated vaccines, where some of the immune modulating genes are inactivated by recombination, effectively causing a less virulent virus. This seems to be, at this stage, probably the leading candidates for vaccines against African swine fever, but as I mentioned, has significant safety concerns associated with live attenuated vaccines at present. Recombinant subunit vaccines have been developed, both protein-based vaccines as well as viral vector vaccines, and some of the newer viral vector vaccines are showing really good promise and circumvents the problem with safety that is associated with live attenuated viruses. And then there are a couple of groups working on replication deficient recombinant African swine fever vaccines, where viral proteins are expressed under control of inducible promoters, something like adenovirus, porcine adenovirus, where you don't have replication of the vaccine carrier, but expression of the viral proteins in situ that then elicits a broad protective response. But most of these vaccines are still in very, very early stage of development. You do find naturally attenuated viruses as well. I'm just showing some of the work that we did a couple of years back with naturally attenuated viruses from Africa, where we used 3 viruses from Africa, one from South Africa, Mkuzi area, one of the areas in the northern parts of KwaZulu-Natal province, a game reserve area. So this virus we know is non-heme absorbing, and at that stage was assumed to be less virulent, or at least at that stage known to cause a subacute infection. Virus from Benin, highly virulent as a control, and then a Mozambican virus that we were not sure as to what the virulence of that virus was. This was compared to one of the vaccine candidates that was developed in Spain, OERT388, and has formed the basis of many of the live attenuated vaccines currently under investigation. So if you look at the disease development post-infection using these different viruses, with OERT completely avirulent, causes asymptomatic infections, no clinical signs, no detectable viremia, seroconverted within 7 to 14 days, and the pigs were terminated at the end of the trial without any significant deterioration in health. Compare that to the Benin virus, which is highly virulent, clinical signs developed within 2 days, viremia within 3 days, seroconversion within 7 days, and death within 7 days of all of the pigs infected with that virus. Very similar, if not worse, situation with Mozambique, the virus from Mozambique, with all of the animals dying within 6 days post-infection. Mkuzi was a slightly different situation. Clinical signs was delayed 7 days. The viremia was only detected at day 18, but seroconversion was seen quite early. And these animals were actually euthanized 18 days after infection when they showed severe signs of ASF, showing that even if you have a delay of onset of disease, the outcome is generally the same with naturally attenuated viruses. This We went on to use this in a vaccine study. And again, not to spend too much time on this, if you look at the solid line, the animals that were not vaccinated all died within 7 days post-infection. We administered either a single dose of the vaccine or a second dose of the vaccine, a booster dose. And survival rates were not much better using these vaccines, slightly better with a delayed onset of disease. So this shows that using naturally attenuated viruses as a basis for further development of live attenuated vaccines does show promise, but additional work is needed to really make the vaccine safe for use in naive pigs. I just quickly want to show also work done by Dr. Linda Dixon and her group at Burbright, using live attenuated vaccines. Dr. Dixon's group was probably one of the first groups that worked on live attenuated vaccines and have done a lot of work on this. And what I want to show here is the cross-reactivity and how difficult it is to actually predict cross-reactivity or cross-protection using live attenuated viruses. So what they did here was they vaccinated animals using viruses were generated originally from the Benin virus, and then challenged with Uganda or with Benin. And you will see that when you use the vaccine and challenge against the same virus that the vaccine was developed against, you get 100% protection, shown in the top graph on the right-hand side. When you challenge with a different virus, so in this case challenging with Uganda, you still get very good protection. But on the left-hand side, they then tested the activity of macrophages isolated from these vaccinated animals against a whole list of different viruses, and you will notice that the Malawi virus indicated in the light blue color on the graph, there's almost no cross-reactivity against that virus, suggesting that should you challenge the vaccinated pigs using the Malawi isolate, it will not be able to protect the pigs against challenge with that virus. But for some of the others, really good protection and really good cross-reactivity with the vaccine. For subunit vaccines, just very briefly, they induce antibody responses, specifically if you use them as protein vaccines. A lot of work was done on this in the early 2000s, specifically using P72, P30, and P54 as targets. But unfortunately, although they delay the onset of the disease, you still have death after challenge in vaccinated groups. Some of the pigs recover and clear the virus, but generally it follows a very similar pattern to what you see with naturally attenuated viruses that I showed. CD2-like protein or the hemagglutinin protein was also used with very similar outcomes. For viral vector vaccines, as I mentioned, the difference with these being that you use a virus to express the protein of interest in vivo. They largely induce CTL responses and have shown very good promise in challenge experiments, with some cases reporting 100% protection in these studies. And very importantly, they reduce the safety concerns associated with live attenuated vaccines. Which brings me now to the really interesting topic, is where are we going with these vaccines and what will happen when they go into the field. Now, after the introduction of African swine fever into China, there was always a concern that considering the number of pigs that is present in China, that the virus might actually mutate and that new strains of the virus would emerge in China due to the epidemiology of the disease in that country. In the beginning of this year, a report was published of the emergence and prevalence of a naturally occurring low virulent virus in domestic pigs in China. The paper describes 22 African swine fever viruses from China that were characterized, 2 of which was non-heme absorbing and had low virulence. Unfortunately, both of these viruses also showed high transmissibility and resulted in non-lethal chronic disease and persistence in infected pigs. When they sequenced the virus, they realized that one of the genes were deleted, the EP204 gene, which encodes the CBD2 protein, which has previously been used as a candidate for live attenuated vaccines. So the consequence of this, it's not clear at that stage, it was not clear whether this was a naturally occurring variant of the genotype 2 virus or whether this was caused by the use of an illegal vaccine. But the fact that this virus so closely resembled viruses that's previously been used for live attenuated vaccines did raise concerns that illegal vaccination was taking place in China. This led to these type of reports in the media, very alarmist for good reason, and some of the leading experts on African swine fever and the use of vaccines, including Professor Vascaino, one of the ASF experts in Europe, warned that illegal use of vaccines endanger the eradication of African swine fever in Asia. The reason being that once we have a chronic infection in Asia or anywhere else, it is very difficult to distinguish between virulent and avirulent infections and to apply traditional control programs to either eradicate or limit the spread of the disease. The situation came to a head earlier this year when another paper was published by the same authors of the first, confirming that a low virulent genotype 1 virus emerged in domestic pigs in China. Again, these viruses were non-heme absorbing and very closely resembled viruses that were previously used for vaccine development. On the right-hand side of the slide, I show a phylogenetic analysis of phylogram of these viruses compared to some of the other genotype 1 virus, and you will notice that the 2 viruses most closely related to these variants in China is OERT-883, which I mentioned before, which was a vaccine candidate, as well as NH68, which is a vaccine candidate developed in Portugal. This is highly suggestive that illegal vaccines have been used in China, and similar to the genotype 2 viruses, these viruses result in chronic infections after they infect pigs. So at first, the pigs will appear normal. They will not develop any clinical signs, but after time, the following clinical signs start to appear as the chronic infection increases. So typically, you would see necrosis on the flank and the abdomen of these animals, significant necrosis, skin necrosis around the head and eye areas, as well as the ears, swollen hocks, a typical sign of arthritis that develop in these pigs, and then similar in this one, just necrosis on the back of these animals. These animals deteriorate to the point where you cannot put them into market and have no other option but to euthanize pigs that are chronically infected with these vaccine strains. So what is being done about this? There are international efforts currently underway to monitor African swine fever variant strains, whether it's natural or through the illegal use of vaccines. So the OIE and FAO has developed, together with some collaborators in China and at USDA in America, diagnostic assays that are able to detect vaccine variants quite effectively. The first uses a triple fluorescent PCR method where we target the P72 gene, which should always be present, but the CD2V and multigene family genes that are targeted by this PCR, if they are not present, it's indicative of a low virulence strain, either through natural infection or illegal use of vaccines. There's also a second protocol that has been developed targeting one of the other vaccine candidates that's currently undergoing clinical trials in Vietnam. This was developed by the USDA and is a deletion of I177L gene that is very indicative of vaccine as well. So these are now available and being used by reference laboratories across the world to monitor the emergence of new variants. Unfortunately, most of these tests require, again, specialized equipment and access to real-time PCR. But it is also, both of these assays will be available in our laboratory at OVI as a reference laboratory within the next couple of months. This just summarizes the interpretation of it. So just as an example, ASF CD2V gene and multigene family double deletion, if you don't have positives on that, it might be indicative that a legal vaccine is being used in your country. Just following on from the international initiatives, the FAO is currently working on updating their diagnostic manuals. We expect to release this by the end of December of this year, which will include updated surveillance tools, both for molecular testing as well as pseudosurveillance to address the emergence of low virulent viruses. The OIE has also established OIE reference laboratory network in Africa. There are 4 representatives from Africa that form part of this network with the intention of expanding the diagnostic capacity in Africa and providing diagnostic support, laboratory support to regional groupings. We're also developing an open access information sharing platform for the dissemination of African swine fever epidemiological information as well as genomic information. As Edward indicated, whole genome sequencing is becoming increasingly important in tracing outbreaks, and specifically as it relates to the use of vaccines or the emergence of natural variants of these vaccines. I also just want to finally end off by saying that the OIE reference laboratories and regional laboratories are there to support field activities. If you ever need the support of the laboratories, please contact us. We are always more than willing to assist in any way possible to contribute to the control of African swine fever on the continent. And with that, I will then conclude this presentation. Thank you.
Thank you, Livio and Edward. Dear colleagues, it is our turn to consolidate, to organize ourselves so that we can get into a discussion. We have 5 minutes until the break. We are going to take a break, but before that time, I don't know, 5 minutes is it's quite some time. I think some of those questions which are on the chat boxes, we can look at them, and once we expend those 5 minutes, we can go for break. Over to you, Livio.
Thank you very much, Thomas. Now, I agree with that. Edward and I will go through the chat now and summarize some of the questions. And then after we come back from the break, we'll start the discussions. So thank you very much for your attention, and we'll be back shortly. Thank you.
I'm trying to check through if I can see any hands, any more chats. The chats are being brought together. I mean, the questions which you put on the chat box, so that it can lead. Yeah, I know. Dear colleagues, maybe it's time to call the room into order. We are going to resume. I just wanted to give for the session, the remaining session up to the conclusion of today's session, we are going to resume and get into a 15 minutes discussion. Then thereafter, we will go to for 10 minutes where the trainers we had today will guide us through the tasks which we are going to undertake by the participants, which are going to be presented next week. And then we will hand over to the trainers who were in the first session, who I know have had a chance to look at the completed questionnaires by the participants from the different countries I had mentioned. At least 5 countries brought in their complete questionnaires. Then we give them 20 minutes. Mary Lou will lead that session, 20 minutes to look at those completed questionnaires and give her view on the way we have performed. So without further ado, I want to return the ball back to Livio so that he can guide through the discussion session. Over to you, Livio.
Thank you very much, Thomas. We've gone through the chats. I could only see one question, and it's a question that we quite often get asked, and that relates to the shipment of samples from countries to either the reference laboratories or I think even to regional laboratories. The shipment of samples is always a concern, not only in Africa. We have very similar situations in the Americas as well, where shipment of samples are really difficult. From our perspective as one of the OIE reference laboratories, I can maybe say that we offer, I'm going to start off actually on the other side of it, the diagnostic aspects of it. The confirmation of outbreaks or the first sample submitted by a country for confirmation of outbreaks, we test without charge. So should any of the countries request us to confirm an outbreak or have a suspicion of an outbreak and send samples to us, we would test that without charge to the country. That is part of our OIE mandate. We have funds available to cover that, and we really do want to encourage all countries to make use of this offer if you can get the samples to us. In terms of shipment of samples, really the problem there is twofold. Firstly, in terms of cost, shipment of infectious material is really expensive in general. But secondly, also logistically, we struggle to find service providers that will work throughout Africa, put it that way. So we have been in negotiations with several of the courier companies in this regard, trying to find ways around the logistical constraints that there is, but unfortunately that has been fairly unsuccessful. What we have done is we have developed different transport protocols that can at least address the problems of spoilage during transport. Because even if we do get a company to transport samples, quite often it takes several days to actually transport those samples from the country of origin to a laboratory, and during that time the samples spoil. In the case of African swine fever, that's not too much of an issue. Edward alluded to the stability of the virus. Purification generally doesn't stop us from even isolating the virus. It makes life a little bit difficult for the laboratory staff, but it's definitely not impossible. And all of the molecular testing that we do can deal with quite significant degradation of samples. So that is not so much of an issue. It's really finding safe ways to transport the virus across borders so that we don't spread the infections further than is the case is already. So from our perspective, I wish I had a simple answer to this. In terms of costs, depending on the situation, the OIE reference laboratory will see whether we can cover the cost, especially early in the year, just after our budgets are released. It is possible for us to contribute to the cost of the transport, but we need to find solutions for the logistics of getting the samples to not only the OIE reference laboratories, but as I mentioned, the regional and even national laboratories that provide support to the veterinary services. I don't know, Edward, if you want to maybe add to that from an East African perspective.
No, nothing much to add. I think that's correct. You've identified the constraint as it is. Thank you.
I can see a hand from Patrick. Would want to say something. I also want to invite the other technical partners who are in the room. Maybe they have something to say, like FAO. I can see Fazina looking at me directly. He could be having something to add. So let me give it to Patrick to say something.
Yes, thank you. My question is also about how FAO could assist. I remember that Akiko Kamata in FAO Rome was tasked with assisting countries on these matters, never mind whether it's for African swine fever or other diseases. But I'm not sure what the status is of this FAO service. The question, of course, at the end of the day is, why would Sierra Leone want to send samples all the way to Pretoria? Is it because they cannot diagnose the disease themselves in Freetown? Because in any case, the services that Pretoria is going to offer is very advanced diagnostics, molecular diagnostics. Maybe that is not what you need. Maybe you just need a simple Penside test or a simple ELISA test to confirm that you're dealing with African swine fever, to the extent that your clinical signs are not enough. And then it's really not worth your while sending samples all the way to Pretoria. It may be simpler to send a test kit to Sierra Leone. And I'm just suggesting this possibility as well. So, but maybe the colleagues from Sierra Leone can clarify where exactly their constraints are.
Sierra Leone, you want to clarify?
Come again? I did not hear the question.
You heard the comments which were given by the reference laboratory and even my colleague Bas saying, do you really require to transport samples all the way to reference laboratory in South Africa from Sierra Leone? Or would you want to get a test kit to do it, to change it another way? And so we wanted a clarification from you.
Yes, initially, we had our laboratory expert from Kenya working for FAO at the Center of Veterinary Laboratory. So what we did, test was conducted at the lab and it was proven positive. But you know, sometimes when you test in our lab, you need the reference laboratory to confirm that what you have done is correct or you have to improve. So shipping this now for confirmation, if without the support of FAO, we can't because it's costly. And then this shipping agency here, their costs, honestly, we can't afford all what they are telling us. So we do send, the recent one we sent to South Africa. Even for most of our samples, is either South Africa or one of the reference laboratory, but with the support of FAO. So my question was now, how as a ministry, because since the livestock division is under the Ministry of Agriculture, can the OIE or AUIBI facilitate, because we do have a sporadic outbreak from these slums areas where people keep rearing these pigs. So in case we have to, because normally what's our problem, if you collect sample and then the result delay, next time when you are going for sample collection in that same area, the farmer will not allow you to sample or to even do anything, because they will say, you keep holding on to our results, we don't know. So it will take like a month or a month plus. So the farmer will be waiting. He or she want to know the results. So for us as a ministry who want to send such sample urgently for confirmation, because we can do it nationally. So if you can have support with the test kit, fine, it will be grateful for us, so that we can know, yes, these kits are okay, and then we can do the test in our laboratory. Thank you.
Thank you for Amara for that clarification. If we were expecting our colleagues from AUIBA to be in the room, I don't see any of them. And if there's any other of our partners who would want to make a point of support to that effect, you have mentioned FAO is doing a great job. And if there's anything else, but what if our partners in AUIBA are not here, we cannot pretend to answer for AUIBA.
Sure.
Yeah. We will take it on and hand that question and request from that it came from Sierra Leone. And because we are going to have another session on the 23rd of November, we will see what they have said and share it for all of you, and ask one of them to come and join us. Over.
Thank you very much, Thomas. I just wanted to comment on that. I think Patrick has a very good point. I understand that in some cases it is necessary for the diagnosis to be confirmed by either OIE reference laboratory, ideally, or a national laboratory, but that's partly why we're trying to build capacity across Africa to cut down on the logistics and the time for the samples to get to South Africa. So to have regional reference laboratories, even if they don't have full OIE status, but they are supported by the OIE reference laboratory, which in this case would be us, could facilitate that same function to confirm the outbreaks. That's the first part of it. So we really are trying to build capacity across the continent. And we've been very successful in building capacity across at least the Southern African portions. And I know Ilri has done a lot of work in East and West Africa as well. But on the supply of reagents to national laboratories, that's something that we are also considering and looking into the feasibility of, and is an important function that we could fulfill. One of the challenges that we face quite often is that test reagents are not readily available when they are needed, especially in countries that don't deal that often with outbreaks. These reagents have expiry dates and they are expensive to maintain. So one of the things that we as a OIE reference network is looking at is trying to build the capacity where emergency stocks are kept at the reference laboratories and are made available as the need arises on with a short turnaround time. That's still in very early days of discussions. Obviously, it has financial implications as well, but I think that would also assist countries in diagnosing and identifying outbreaks as they occur. Thank you. I haven't seen Thomas.
Yeah, thank you. You can go ahead.
Now, I was just looking at whether there's any other questions or anything on the chat, and I haven't seen anything else come up. But maybe I can just ask Edward. Edward, you mentioned the different pork products, and in your talk you also mentioned warthog meat and so on. Can you maybe just elaborate on your opinion and experience specifically with bush meats, warthog meat being hunted, and the risk that that poses for spread of the disease?
Yeah, okay. So, yeah, so our hypothesis is that in some of the studies that we did in Kenya and also in Uganda, we could, one of the key features of our diagnostics was that we couldn't pick any antibodies from bush pigs. But when we did a screening of the tissues, we were able to pick up a virus by PCR, and also were able to isolate viruses from them. So the assumption is that the fact that you can isolate the virus, it means components of the tissues from those animals could potentially actually be infectious. So in places like Uganda, maybe if the people from Uganda here would confirm that animals that are outside protected areas are considered as
I think we might have lost Edward there. Thomas, can you just confirm that it's not my connection that's gone?
Yeah, I lost him too.
Okay, thanks, Mary-Lou. Whilst we wait for him to come back, I see there's a question from Namibia. You want to?
Thank you. Thank you very much. For me, I don't know, maybe I got it wrong. There was a sentence in the presentation for Edward that there was a mention of bush pigs, warthog. You know, we are from the south here. To us, bush pigs are warthogs. We just wanted to understand if there is a difference between a warthog and a bush pig. Thank you.
Thank you very much.
So,
sorry, we lost you there for a second.
A different species, or maybe Liv, you can go ahead.
No, please, Edward, you are the expert there, so you're welcome to continue.
Okay. Yeah, so physically and even morphologically, they are different. Warthogs mainly live in burrows. You'll find them under culverts and burrows, but bush pigs nest in bushy areas. No wonder I think they are called bush pigs, and they are different species in terms of if you want to identify them. So so they are a bit different, and if I had pictures, I could have, maybe in the next presentation, I'll share with you the difference between them. So the bush pigs looks more closer to the pig in their feature, to the extent that sometimes we think that they could even crossbreed. But warthogs are quite a bit far from our domestic pigs, and they have this feature of long canines that come out like tusks, which bush pigs don't have.
Thank you. Thank you so much.
Thank you very much, Edward. Mary-Lou, you want to add to that?
Yeah, I'd just like to add to that it's probable that in Namibia you wouldn't have a lot of bush pigs because they like sort of forested area, thick bush, and it's essentially a dry country, and warthogs are much more likely because they live in open savannas, and as I've heard you, they burrow or they live under road culverts. Important thing about warthogs that Edward didn't mention, they've got these big warts on their faces. It makes them look rather different from other pigs because they've got, especially the males, big sticking out warts on their cheeks, and bush pigs lack that. They just look like a sort of a skinny looking or a different looking pig, a hairy looking pig with a long nose. Thank you.
Thank you, Mary-Lou. Patrick, I see you have your hand up.
Yeah, maybe I misunderstood the question from Namibia, but maybe the underlying question was, is there any difference in sensitivity to the virus? Because, I mean, the physical appearance, I think is pretty irrelevant, but is there a difference in sensitivity to the virus between the two species?
Yes, that would
Yeah.
have been a follow-up question. Yes.
Yeah, so for all the wild species of pigs in Africa are resistant to African swine fever virus. So this includes the warthogs, the bush pigs, giant forest hog, red river hogs, all those are resistant. But this is in contrast to the wild pigs in Europe, the wild boars that are susceptible. I don't know whether that helps, over.
Yes, it does. Thank you.
Yeah.
Thank you, Edward. Patrick, is that an old hand or do you have a new comment?
No, I put my hand down. Thank you.
Thank you. No problem. Thomas, I think we're getting to the end of the time allocated to us, if I'm correct. So if there's no further questions, I just again would like to firstly thank Edward for his brilliant presentation and his assistance, and for the opportunity to engage with all the participants. I'm going to hand over then to, oh, sorry, Thomas, the next is our tasks, I think.
Yes, next week's task.
Okay. Edward, do you want to present your task first and then I'll go after you? Just unmute. Edward, you're still muted.
Okay, sorry for that. So what we'll be sending you from my side as a task is we realize that the virus transmission is very critical when you want to analyze a risk of a spread of the virus and also the risk of African swine fever. And so I've set out some points. It's just a list of key points that came from the presentation of some unique features of the virus and some characteristics that I described. And basically throwing it back to the audience that, you know, with this kind of knowledge, can this help you then develop some security messages that you can use to educate farmers? So it will just be a list of this, and we want you to make sure that it is as contextual as possible to your situations in your country. What kind of messages will be more understandable to the farmers that you work with? So this will be sent to you by Dr. Dulu, and then if you have any questions, definitely my email will also be there so that I can clarify. Over, leave it to you.
Thank you very much, Edward. I think it's going to be really interesting to see the feedback from the participants, as you say, in their own context, what they consider and how they would go about developing this information. The second task from this session is specifically on the diagnostics, and I'll just very quickly share my screen again. Here we go. So it relates to decision trees. So all the information from that you require to complete this task is contained within the presentations, which you will receive copies of as well, as Thomas mentioned. But what we effectively want you to do is develop decision trees or complete decision trees for 3 different scenarios that we've identified based on the situation, the epidemiology of African swine fever in Africa. And I'll just give you an example of the first. I'm not going to obviously give you the answers, but just as an example of what we mean. In scenario 1, we say that the pig keeper or pig keepers in a border town reported a large number of pigs dying suddenly with no apparent signs of disease. On inspection, veterinary services find pigs showing some cyanosis on the tips of the ears and bloody diarrhea. Veterinarian suspects that the animals may have died of African swine fever, and it is decided to submit samples for testing. Then you need to complete the decision tree and provide a final conclusion on the cause of death. And what we mean by that is just fairly simple. We've given you a basic outline of a decision tree with certain gaps that you need to fill in. So everywhere that there's a white block, there's an answer required. And based on that scenario, you will then fill in the required information and provide a final conclusion. We've provided 3 different scenarios of this that you can then use to develop the decision trees. A fairly simple task. I'm sure that just following through from the presentations, both mine and Edward's, you'll be able to do this quite easily. So those are the 2 tasks that we've assigned for the present from the session of today. If there are any questions, I'll just maybe quickly open up if there's any questions specifically related to the tasks. If not, then I'll hand over to Thomas. So I don't see anything on any questions on the slides or on the tasks that we've assigned. So we assume that everything is clear and we look forward to receiving your inputs there. Thomas, with that, I'll then hand over back to you. Thank you very much.
There seems to be a question from regarding Edward. Edward, can you not share your task as well? Maybe that makes it easier for people to understand what exactly you expect them to do.
Okay. Okay, shortly. Yeah, can you see something?
Yes, you can just make it slightly bigger, please, Edward. It's fairly small. Just zoom in.
Does that work?
Yeah, that should be fine there, I think.
Yeah, okay. Yeah, so it's the title is African Swine Fever Virus Transmission Task. So the question here is, using the following information on African swine fever and African swine fever virus, formulate 5 biosecurity messages targeting pig farmers in your country. So some of those messages are listed there, and these are basically facts from the presentation that will be shared with you. The fact that soft T colonies can maintain virus for up to 15 months in absence of blood meal, making them capable of transmitting the virus. If you were to formulate a message around this, how would that message look like, for example? And so that's basically the assignment. Any questions?
Thank you, Edward. I think that's a little bit clearer. So it's basically just developing biosecurity messages based on some of the key epidemiological characteristics of African swine fever in Africa.
Yeah.
Thank you. Okay. I think so, yes. So thank you very much. So Thomas, I'm going to hand over to you then now.
Thank you. Thank you. Now it's my pleasure to invite Mary-Lou to guide us through the last week's tasks, which were submitted, and who she's going to choose. I don't know the methodology she's going to use, but now it's over to her, Professor Mary-Lou.
Thank you very much, Thomas. I thought that it's possibly, seeing there were few questionnaires, only 6 countries did the task. I thought that we might just show those on the screen and mention, we can discuss things. It's pretty obvious that the majority of pigs and pork get imported from South Africa. So obviously the risk assessors are going to have to focus on us, but in general, we are fairly careful about what we export. Of course, what goes out illegally, we have no idea. But would that be a way to do this? Because I looked and I thought, well, I suppose I could have made a table to compare the different countries, but the data didn't seem to lend itself. So with your permission, I'm going to show the forms that were filled in, and we can just highlight certain things. And because some of them are very different in the numbers of pigs and the pig production systems and so on. And then I hope that seeing them would inspire some of the other countries, because it's not too late. You can still do the exercise and send it in. It won't be discussed in a session. Now, Thomas, I'm going to try and share my screen. I'm not very good at doing this with Word documents, so I might have to call on you to get into that Dropbox and help me, but let's see if I can do it. Yeah. Is everybody seeing it?
Yes, we are.
Because I can't, there isn't a show mode for Word, so we'll just have to go through it. But Namibia has a small pig population, as you can see, and they're very good about keeping their census up to date. And they have both commercial farming and traditional pig production systems. If live pigs are imported, they mainly come from South Africa. They're not importing any other genetic material. They will import pork from South Africa and Europe, as well as processed pork products. And the processed pork products are processed to destroy the African swine fever virus. I expect you heard some of that was necessary and from Edward's talk in compliance with the articles, necessary article of the OIE Terrestrial Animal Health Code. So they're very careful, and I'll just mention at this point that South Africa, because we're not free of African swine fever, if you've heard, our commercial pigs, it's about 65%, I think, Peter Evans will correct me if I'm wrong, of the commercial herd is compartmentalized. So they are in compartments which are audited. There's a whole protocol for what you need to have before you will even be looked at for a compartment. And they're certified by our veterinary authorities as free of African swine fever, classical swine fever, and foot and mouth disease. And they can therefore safely export their product in the terms of live pigs. But because the OIE obviously can't go around certifying individual farms, it's the government services that certify the farms, and then there's an agreement with trading partners who can look at the information and say, yes, that sounds good enough for us, we'll take the pigs, or we'll take the pork products. The pigs from a compartment are slaughtered at specific abattoirs and that are certified for export, and they're not slaughtered together with pigs from non-compartments on the same day, as far as I know. So there's a fair amount of assurance there. So that was Namibia. Thank you very much for submitting that. Let's get the next one. You can all see this one as well, can't you? I'm still sharing, and we now have the form from Seychelles. Can everybody see it? Thomas, can you help?
Yeah, yes, we can see.
Let's see. Okay. Now, Seychelles has a very small pig population. I'm very happy to tell Seychelles that you have the second smallest pig population of the countries that do pigs in Africa. Botswana has fewer pigs than Seychelles. So you have fatness, and you have intensive and semi-intensive systems. I can imagine that in an island with not a lot of land available, you won't have these sort of free-ranging systems. Pigs are imported from South Africa and from Spain. Semen and embryos would come from the Netherlands, which is a very good pig-producing countries. Pork can come from Brazil, Poland, South Africa, Denmark, and Belgium. And of those countries, Poland and South Africa are not free of African swine fever, but I'm sure they would, as you say, the products that you do, the pork that you import does comply with the OIE requirement. And I'm quite sure that that is the case. Belgium, in fact, had a short period of African swine fever in their wild boars for about, just about nearly a year. But it remained in the wild boars and it didn't spread to domestic pigs. And yeah, not exporting pigs or pork. The next country that complied is Sierra Leone. Now, so far of the countries, you have the largest pig population. It's still not very large, but it's a reasonable pig population. You're not a huge country. So you have extensive, semi-intensive and intensive pig farming. And this interests me very much because I've often tried to look and see what different countries have, and I had concluded that Sierra Leone hasn't had African swine fever or reported this until recently because they only had intensively farmed pigs, but I can see that's not the case. And in fact, Sierra Leone did report made its first report of African swine fever in 2020. So you import live pigs from Brazil, yes. No embryo pork from Brazil, again, yes. And you don't export. So thanks, yes. And Brazil's probably, it's near the top of the exporting countries for pork. They have huge pork, pig population. And a lot of what one can import depends on how easy it is to get there. And Brazil seems to make it pretty easy for you to get your pork. Let's go to the next one. South Sudan. That's amazing because if one goes to the FAIRSTAT database, they tell us that South Sudan has no pigs at all. And I was quite sure that wouldn't be the case. So you have rural, and rural, urban, and I guess that is mostly smallholder pig farming. You can correct me if I'm wrong. You import pigs from your neighbors, Uganda and Kenya, but you don't import pork or export anything. So I find it, I'm very pleased that you responded because it was a country that I knew nothing about. And as far as pigs were concerned, because there isn't any information available on FAIRSTAT, but I was in touch in one of the online web-based master's degree courses that I teach at the University of Pretoria. We had a vet from South Sudan last year, and he informed me that yes, there were pigs in South Sudan. So thank you very much for that. Now we come to the countries with masses of pigs. Tanzania was the next one too. Over 3 million pigs, which is very nice because FAIRSTAT will tell you have just under 500,000 pigs. So I'm glad to see that this is, I knew that wasn't correct from Tanzanian vets, and you have a large pig population. Smallholder keeping 1 to 31 pigs and average very small herd of under 5, and then intensive production systems. And you import from South Africa, Zambia, Burundi, Rwanda, and Malawi. I see that in italics, so I'm going to ask the delegate from Sierra Leone to unpack this a little bit. Yes, Tanzania, you get your small amounts of semen and embryos, so it's not really imported. Only in Zambia, you get, and I see they do come from Zambia, and it's your Pig Farmer Association that imports it, right? That's interesting to know that you are using artificial insemination. And you get your fresh and frozen pork from Kenya and South Africa. Yes, Kenya, Farmer's Choice exports quite a lot of pork to the region. And the processed pork does comply with the OIE code. And non-edible products as well. And then exporting, I see. Now, officially you don't export pigs, but they're shared along the border, especially Malawi and Burundi. And so I guess that you put the italics in question 4, the Burundi, Rwanda, and Malawi, because you don't officially import pigs from those countries, but along the borders it can happen. Am I right?
Yes, you are right.
Thank you.
Yeah,
yes.
that's why last week. Yeah, that's why last week we asked you if it's official and unofficial, because they share.
Yeah. Yeah, thank you very much for that, for asking that. And I said, yes, if you know about unofficial, we'd love to know about that too, because I think that does have to be factored into any risk analysis. And there are many of the countries, Namibia, I think, shares a pig population with neighboring Angola up on the northern border. West Uganda and Kenya certainly share a pig population in the west, which Edward, I think, mentioned. And Zimbabwe and Mozambique share a bit of a pig population, or at least share African swine fever outbreaks. So yes, thank you for telling us that. And that is interesting, and it's great to know that you do know about that. And finally, we get the most pig-rich country who responded, nearly 4.5 million pigs in Uganda. And you have intensive, semi-intensive, and free range. You do import live pigs from Belgium, Kenya, Rwanda, South Africa, and Zambia. That's quite a selection. You do import semen from South Africa, and pork from Kenya, South Africa, and Belgium. And processed is safe. No non-edible products. And you say you do export pigs. Yes, obviously to your neighbors. And pork, you can export to Vietnam, DRC, South Sudan, India, Hong Kong, Kenya, and China. That's quite a good selection, and it's clear that your veterinary authorities can certify very well, because Uganda does have African swine fever. But again, I know that you have some very good commercial farms. I also know that the one great ambition I have to do in my life again is to go and eat, once again, eat pork in a pork joint in Kampala. It's a wonderful experience. And thank you very much. I'm now going to stop the screen share. And has anyone got any questions or comments, or would anybody from the countries that didn't supply us with any information like to tell us briefly what they would have filled in on their form, or would you like to fill in the form later? It's over to you, to the floor, to come up with. I don't know, let me check the chat room.
There is a hand from Swatini.
Please go ahead, Swatini.
Yes, thank you, Marilu. For me, it's not a question per se. It's just to say Swatini did submit the questionnaire, but I didn't see you present it there.
I didn't present it because I didn't receive it. Could you send it again, perhaps? Did you send it?
We submitted to Dr. Thomas Dulu, and he did acknowledge receiving it.
Okay, it's not in the Dropbox with the others. I don't know. Did it come in quite late, Thomas?
Yeah, maybe there are some, one of the ones which came quite late. We'll put it in the Dropbox. Thank you. Thank you for submitting.
Thanks very much for telling us that and for submitting. And yeah, thank you very much. And what I'm going to do is I will compile a table when I've got more forms in, and that can be put in the Dropbox and circulated to all of you. Thanks for that. Sierra Leone?
Yes, thank you so much for what you just presented. So for us, this number you are seeing, it was like the head count that was done in 2019 prior to the outbreak. So it was just for Freetown. You know, currently we have people, yes, this number is just for Freetown. We have currently people rearing pigs all over the country in the interior, not only the ears. So we didn't capture that one because the ministry did not yet plan, because it was, we were in the planning of a mini survey or a census for animals, but like the support was so minimal. So we started with the slums, because in Freetown here, most of these pig rearer are on the alongside the slums area, I mean towards the sea. So when they have problem, they do call us. When we go for treatment, we ask them how many you have. So per farmer, this is the exact number we have put. But this is from 2019. So we know that number may be an increase now from 2019 to 2021. So that's just the clarification I wanted to make.
Thank you very much. Thank you for that. Yes, I expect you'll have, it's quite difficult to know where all those pigs are. I can share with you now that South Africa's pig figures in the Vrystaat and that I know are to a very large extent apply to our commercial industry, and we're not as well informed at all about what happens in the rural areas, even the peri-urban areas. Thank you very much. Sharon, would you like to go ahead?
Thank you, Mary. Mine is just a comment looking at the presentations from Seychelles and South Sudan. I just wanted to comment, maybe the volumes are very low, but we did export about 42 tons of products into South Sudan in 2020, and about 36 tons into Seychelles. So just a comment there, but I noticed that that wasn't captured. So I wonder, maybe the guys from those countries can comment on that or something. Thank you.
Thank you very much. Would Seychelles or the South Sudan like to comment on that? No comments. We can update the information accordingly. Thank you very much, Sharon. Anyone else like to comment or ask something? If not, then I think I should hand it back to Thomas.
Thank you, Mary Lou, for that summary. I think we have gone full scale, and I also picked the input which came from Eswatini and put it on the box. And if there are any others, we are still opening for them to send their questionnaire, completed questionnaire. There's some vital information which is coming out, which nobody has ever known about. And even the ones who have not given all the information, it is important that it is preparing ourselves for the next the risk analysis training, because all the data sets you have are very important for what we are going to get into and to get trained. So with, I can say, Professor Merilu, I give back to you again.
Sorry, I have just found the Eswatini questionnaire, and it's been done very nicely. So perhaps I could just quickly share it with everyone. Would that be okay?
Sure.
Okay, I shouldn't take too long. So Eswatini has a small population, fairly small, a middle-sized, I'd say, for Africa population. And they have pigs that are housed with high biosecurity, 20%, with 70% have biosecurity and 9% no biosecurity and 1% free range. So they have the whole range, and it's really nice that they seem to know what they've got. And they do import live pigs from South Africa, from the compartments, and as well as semen or embryos of pigs from South Africa and sometimes from the Republic of China, Taiwan. And Taiwan, I'm happy to say, has so far managed to not have African swine fever, in spite of the fact that mainland China has it in a very large measure. And oh, you've stopped importing fresh pork. I expect you can get enough from those 50,000 pigs to feed yourselves. And you do import processed pork products, which have been processed according to the article of the OIE, Terrestrial Animal Health Code, and no export. So thank you very much indeed for that. That's good information to have. And yeah.
Thank you, Professor Merilu. I think we have come to an end. I'm sure my colleagues in Swatini, they wanted to see their whatever they submitted to us, so that we can show everybody what they have brought in. And to do that, I think let me open and so that I can share.
Yes, sorry, I didn't actually get to sharing it.
Okay.
That's great. That's great.
You can see it?
Yes, it's clear, visible.
Yeah. And this is the comments which are coming from that table. And so I think they did justice, and like we just said. Okay. I think we have reached a point that we have wanted to, we should have called it a day 10 minutes ago. And it's been very rigorous. What I'm going to do after we close is to send you the presentations which we had today, PDF format, and then the tasks which were given by the two trainers for today's session, so that we prepare for next session. Next session will be on the 23rd of November. That is Tuesday next week. So I think this is where I just wanted to end. I don't know if there are any of our observers who would want to say a word today. I could give them a chance just to say one word. A minute only. None?
Yes, Thomas, I'd just like to thank again the team that's doing the training and the presenters, and really also thank the participants. This is very, very commendable. And I'm really happy to see the enthusiasm and the response that are coming, giving new information. And we look forward to your next meeting next week, Tuesday. Thank you.
There's a hand from Martin, Argentina.
Well, thank you, Thomas. For us, it's really nice to see the situation and how we are managing ASF. Just to let you know that in our continent, one of the actions we are working on in our region is that countries have the capacity, let's say, to understand how their country is. So for us, it's quite relevant that countries know more about their peak population, about their population dynamics. And that's the base of a risk analysis. And that's just the ideal way just to deal with an introduction of an exotic disease and also for an existing disease. So we are going in the same path with different epidemiological situations, but we are focusing also on that countries have to be empowered, having the capacity to have their own risk characterizations and their own risk analysis. Thank you.
Thank you, Martin. And session 3, that is coming on 23rd of November. It will be overview of value chains associated with pig production and marketing in Africa. Very, very important. It's going to be data sets and information which we have never had before. So it is important that you come in and we share, and with your tasks completed, it will never be the same again. With that, I want to close today's session and thank you all for participating and being very loyal trainees and participants for this training. With that, I want to say bye-bye.
Asante, kwaheri.
Okay, goodbye.